So far, the energy from the light reaction is stored in the chemical bonds of ATP and NADPH. In the Calvin cycle, this energy is used to produce sugar molecules. The Calvin cycle is a complex series of chemical reactions carried out in the stroma. The Calvin cycle begins with the input of carbon dioxide. 3 molecules of carbon dioxide are added to 3 molecules of a 5-carbon sugar called RuBP. These molecules are then rearranged to form 6 molecules of 3-PGA, which has 3 carbons each. In the next two reactions, products from the light reaction are used to boost the energy in these carbon molecules. First, high-energy phosphate groups are added. 6 ADP molecules contribute phosphate groups so that each carbon molecule receives an additional phosphate group. Electrons are added in the next reaction. 6 molecules of NADPH are oxidized. The electrons from NADPH reduce the 6 3-carbon compounds, creating 6 high-energy G3P molecules. G3P is a sugar, the final product of the Calvin cycle. 1 G3P molecules represents the 3 carbon dioxide molecules fixed so far. The other 5 J3Ps are reshuffled to regenerate the original RuBP molecules. To summarize, so far the Calvin cycle has used the energy of the light reactions to reduce 3 molecules of carbon dioxide and produce 1 molecule of G3P. 3 more molecules of carbon dioxide are fixed to form G3P in the same complicated way. To make each G3P, the Calvin cycle consumes 9 ADP molecules and 6 NADPH molecules. These are regenerated in the light reactions. G3P is the actual final product of the Calvin cycle. The cell can combine 2 G3Ps to make glucose, which stores the energy that chlorophyll originally captured from the sun. Factors that affect photosynthesis. 1 is light intensity, 2 is carbon dioxide concentration, and the third is temperature. Organisms extract energy stored in glucose molecules. Through the process of cellular respiration, cells mix the carbon in glucose into stable carbon dioxide molecules and produce energy. Thus, stable and less stable compounds alternate during the carbon cycle and provide a continuous supply of energy for life processes in an ecosystem. In the first stage of cellular respiration, glucose is broken down in the cytoplasm by glycolysis. Glycolysis is an anaerobic breakdown of glucose to pyruvate, which makes a small amount of energy available to cells in the form of ATP. 2 ADP molecules are used to break glucose into 2 smaller units. A phosphate group is added to the 6-carbon compound. Each 3-carbon compound reacts with the phosphate group. Hydrogen atoms are transferred to NAD+, producing NADH. Each 3-carbon sugar is converted to a 3-carbon molecule of pyruvate. 4 ATP molecules are produced. There are 2 types of cell transport. One is passive transport and the other is active transport. In passive transport, substances cross the cell membrane down their concentration gradient. Some substances diffuse through the lipid bilayer, others diffuse through transport proteins. Small nonpolar molecules can pass directly through the lipid bilayer. This type of movement is called the simple diffusion. The animation that you can see here is a simple diffusion. Many ions and polar molecules that are important for cell function do not diffuse easily to the nonpolar lipid bilayer. During facilitated diffusion, transport proteins help these substances diffuse to the cell membrane. There are 2 types of transport proteins, channel proteins and carrier proteins. Many substances, such as proteins, are too large to be transported by carrier proteins. Instead, they cross the cell membrane in vesicles. Exocytosis is the movement of material out of the cell by means of a vesicle. Endocytosis is the movement of a large substance into a cell by means of a vesicle. Active transport requires energy to move substances against their concentration gradients. One of the most important carrier proteins in animal cells is the sodium-potassium pump. Sodium ions inside the cell bind to the carrier protein. A phosphate group from ATP transfers energy to the protein. The protein changes its shape and releases the sodium ions outside the cell membrane. Outside the cell, potassium ions bind to the pump. As a result, the phosphate group is released from the pump. The pump returns to its original shape and releases the potassium ions inside the cell membrane. For every 3 sodium ions taken out, 2 potassium ions are brought inside. As the body of the multicellular organisms grow larger, its cells do not grow large. Instead, the body grows by producing more cells. New cells are needed to help tissues and organs grow. New cells also replace damaged cells. Each new cell also gets an entire copy of the cell's DNA. The large molecule of DNA is organized into hereditary units called genes. A gene is a segment of DNA that codes for RNA and protein. DNA is organized and packaged into structures called chromosomes. A prokaryotic cell has a single circular molecule of DNA. The chromosome is condensed through repeated twisting or winding, like a rubber band twisting upon itself many times. Eukaryotic DNA is packaged into highly condensed chromosome structures with the help of many proteins. The DNA and proteins make up a substance called chromatin. The first level of packaging is done by a class of proteins called histones. A group of 8 histones come together to form a disc-shaped histone core. The structure made up of a histone core and a DNA around it is called a nucleosome. Each of the 2 thick strands, called a chromatid, is made of a single long molecule of DNA. Identical pairs, called sister chromatids, are held together at a region called the centromere. During cell division, the sister chromatids are separated at the centromere, and one ends up in each daughter cell. This ensures that each new cell has the same genetic information as the parent cell. The cell cycle is a repeating sequence of cellular growth and division during the life of a cell. The cell cycle is made up of 5 phases. The first 3 phases together are known as interphase. The remaining 2 phases make up cell division. During interphase, the cell is not dividing. Each new cell requires a complete set of organelles, including a nucleus. The process of dividing the nucleus into 2 daughter nuclei is called mitosis. The process of separating the organelles and the cytoplasm is called cytokinesis. During the cell cycle, a cell undergoes an inspection process to ensure that the cell is ready for the next phase in the cell cycle. There are 3 main checkpoints in the cell cycle. You can refer to the diagram to see it. And finally, we're done. I hope you learned something.